and Harrison in 1907 is the one who cultivated the frog nerve cells in lymph node held by the hanging frog technique. So as like that, there are major developments in cell culture technology. So the first development that was the use of antibiotics, which inhibits the growth of contaminants. And the second was the use of trypsin to remove the adherent cells from the subculture, further from the culture phase. So third was the use of chemically defined cell culture medium. Why we need the cell culture? Why the cell culture is used for? So the areas where the cell culture technology is playing a major role. The model organisms, this acts as a model system for studying the basic cell biology and interactions between the disease and disease-causing agents and the cells, effects of drugs on cells, and the process and triggering of aging and nutritional studies. And we can also use this technology for testing the toxicity test for to study the effects of new drugs and cancer research, study the function of various chemicals, viruses, and radiation to convert normal culture cells to cancer cells. And when coming to the virology, genetic engineering, instead we discussed all this. So we'll go with the animal cell. Okay, animal cell. Can anyone please answer that my screen is sharing?
Ma'am, it's still not visible, ma'am. It's loading.
Are you able to see my screen?
No, ma'am.
No. For me, it is something like your screen sharing is paused. That's why I am asking. Okay. I already started my screen sharing. Why? Yeah. So, animal cell culture that is basically involving in vitro in the laboratory maintenance and propagation of animal cells in a suitable nutrient. So, as we discussed in the 1907, Ross Harrison is the one who developed a frog tissue culture technique. In 1940s, chick embryo tissue that became a favorite for culture techniques. And the interest in culturing the human tissues that started in the year of 1950s after it was demonstrated by Hella Gay that human tumor cells that could give rise to continuous cell lines. Among the various animal cell cultures, mouse cell cultures are the most common used in the laboratory. So, some terminology that involved in the cell culture is organ culture, the culture of native tissue, here undesegregated tissue that retains most of the in vivo histological features, uh, which is regarded as organ culture. Means here we are using any organ. Cell culture, the cell culture, in the cell culture technique, this refers to the culturing of dispersed, means disaggregated. Here in organ culture, it is saying that it is a native tissue and disaggregated tissue. In cell culture, here we are discussing the disaggregated cells that are obtained from the original tissue or from a cell line. Histotypic culture, the culturing of the cells of their reaggregation to form a tissue like structure represents the histotypic culture. And organotypic culture means this culture technique that involves the recombination of different cell types to form a more defined tissue or an organ. Here this picture saying that how the organ culture looks like and the expand culture looks like and the dissociated cell culture and organotype culture. So the first picture, see here, the tissue at gas liquid interface, the histological structure that are maintained by this manner. And the expand culture, here you can see, yesterday I have shown you the culture bottle. So a tissue at a solid liquid interface cells that migrate to form the outflow. And dissociated cell culture, the disaggregated tissue cells form monolayer. Here you can see the formation of the monolayer in this picture clearly at a solid liquid interface. And organic culture, the different cells that co-cultured with or without matrix, the organotype structure that recreated. So when coming to the primary culture, here the culture that produces by the freshly isolated cells or tissue taken from an organism is the primary culture. So the culture which produced by the freshly associated cells or the tissue that is taken from an organism is the primary culture. These cells that are heterogeneous and slow growing and represent the cell or it represents the tissue of their origin with regard to their properties. When coming to the cell line, yesterday I have explained you how the primary culture looks like and finally how the cell line is produced. So here the cell line, the subculturing of yes, Jerusha. Even I am also trying, but he's saying that my screen is paused.
Okay, ma'am.
Shall I stop and share again? Still it is showing the same. Otherwise, I can do one thing. I'll share this PPT with any one of your classmates. Then they will share. Okay, Javish. So the subculturing of primary the subculturing of the primary culture that gives rise to the cell line. The term continuous cell line that implies the indefinite growth of the cells in the subsequent subculture. On the other hand, the finite cell that lies that represent the death of the cells after several subcultures. Yesterday I have shown you what is finite cell and what is definite cell, and what is indefinite growth. Everything we discussed yesterday, no? So why we need the subculturing? Once the available substrate, the surface, is covered by the cells, that condition is usually called as confluency or confluent culture. Means the appropriate culture. So once the available substrate, that surface is covered by the cell, that growth, the slow becomes, the growth becomes very slow, and somehow the growth becomes like ceases. Once it will reach a stage. As everyone knowing, lag phase, log phase, stationary phase, and decline phase. So now we are talking about the log phase, means the uh, substantial growth of the cells. So soon after reaching to the substantial growth, it reaches to stagnant growth, means stationary growth. So where the growth becomes like very slow, thereafter the growth is ceases, then it will reach us to the decline phase. The cells to be kept in healthy and in growing state have to be subcultured or passaged. Means the cell which is there in a stage like log phase means healthy stage. So by the time we have to take the healthy grown culture, means cells, and we have to make it like subculture. This technique is also called as passaging or passage. It is the passage of the cells when they reach to the confluency about 80 to 90% in the flask, disc, or the plate. So here for making the subculturing or passaging, we need the enzymes. Those were like trypsin, dipase, collagenase in combination with the EDTA. The EDTA which breaks the cellular glue that that the cells to the surface, which attaches the cells to the surface. Sunita, now animal biotechnology class is happening. Was you there yesterday? It is the continuation class for yesterday. Clear?
Good morning, ma'am. Can you please let me know what topic is being taken so that I can go for the reference in the book?
Sorry.
Can you let me know what topic is being taken so that I can go for the reference in the book, ma'am?
Are you there in the yesterday's class?
No, ma'am. Yesterday I have missed the class. That's why I'm asking which topic is going on.
Okay. Actually, now we are discussing about the cell tissue culture.
Yeah. Thank you,
and
ma'am. Thank you so much.
preparation of culture.
Yeah. Okay. Fine, ma'am. Thank you.
So facilitates for animal cell culture. Here, while designing the laboratory for animal cell culture technology, the utmost care should be taken with regard to the maintenance of aseptic conditions. So here, the facilities that required regarding the infrastructure and equipment is the clean and quite sterile area. And here I wanted to note you people that the one who already attended my offline classes, they will follow this topic easily. because we already discussed with this. We already discussed the same topic previously. So clean and clear and quite sterile area we need for cell culture, and the preparation facilities and animal house and microbial laboratory and storage facilities like for glassware, chemicals, liquids, and small equipment. All these are the minimal requirements for cell culture. Along with this, we require the equipment like laminar airflow, sterilizer, incubator, and refrigerator, uh, and the freezer like minus 20 degrees and minus 80 degrees. And we need the balance, CO2 cylinder, centrifuge, the inverted microscope for observing the cells, and water purifier and hemocytometer for cell count, and liquid nitrogen for freezing the cells, and slow cooling devices for freezing the cells, and the pipette washer and deep washing sink, because we already dealing with the cell culture, no? That is what we need, the deep washing sink. Besides this, the basic and minimal requirements, there are many more facilities that may be beneficial or useful for tissue cultures, that animal tissue cultures. These include like air conditioned rooms and containment room for biohazard waste disposal and biohazard work doing, and the phase contrast microscope and fluorescent microscope and confocal microscope for checking the stages of the cell lines, and high capacity centrifuge, and this is for and the other thing is time lapse video equipment. Along with us, along with these, we also need the culture vessels. Here in the tissue culture technology, the cells that attach to the surface of a vessel, which serves as the substrate and growth. Hence, there is a lots of important to the nature of materials that are used and the quality of the culture vessels. The term anchorage dependent cells is used when the cells that require an attachment for their growth. On the other hand, some cells undergo transformation and that become anchorage independent. So as just we discussed about the materials that are used for the culture vessels, like glass. Although glass was the original substrate used for the culturing, it is used is almost discontinued now. This is mainly because of the availability of suitable and alternate substrates. And yes, disposable plastic. Nowadays, uh, many laboratories are encouraging the disposable plastic and synthetic plastic material with good consistency, and the optical properties are now in use to provide the uniform and reproducible cultures. The most commonly used plastics are polystyrene, polyvinyl chloride, PVC, and polycarbonate, and metinates, and thermonates. Thermonates means TPX. And types of culture vessels. Here we have different types of culture vessels that we have already uh, had a look yesterday, like multi-wall plates, petri dishes, flasks, stirrer bottles. Uh, Here you can see the requirements like substrate or liquid, cell culture flask, and scaffold materials like multi-wall plates, petri dishes, flasks, stirrer bottles. And what is the advantages, utmost advantages of this technique? Means that tissue culture is the control of physico-chemical environment like a pH, temperature, dissolved gases like oxygen and carbon dioxide, osmolarity, and regulation of physiological conditions on nutrient concentrations and cell-to-cell interactions. And here we can able to control the hormones and hormonal control. The cultured cell lines that become homogeneous, like cells that are identical after one or two subcultures. This is in contrast to the heterogeneous cells of tissue samples, the homogeneous cells that are highly useful for a wide range of purposes. It is easy to characterize the cells for cytological and immunological studies. And we can able to store the cultured cells in liquid nitrogen for several years. If we will passage and if we will store in a proper way in -20, after that, if we will transfer the cells after keeping in the liquid nitrogen and storing in the -80, yes, that remains same if we will keep in the DMSO, because DMSO that arrests the cells. So due to the direct access and contact to the cells, the biological studies that can be carried out more conveniently. And the main advantage is the low quantities of the reagents that required in contrast to the in vivo studies, where most of the reagents, about more than 90% cases, that are lost by distribution to various tissues and exertion. Utility of tissue cultures will drastically reduce the use of animals for various experiments. So these are all all these various advantages we have in tissue culture. So where there is an advantage, there will be a limitation. So what are the limitations of what tissue culture means? Yes, definitely there are several limitations of tissue culture we have. So someone has to expertise and technical skill for the development and regular use of tissue culture. So there will be a cost factor, which is a major limitation for the establishment of infrastructure, and equipment and other facilities are expensive. And it is estimated that the cost of production of cells is about 10 times higher than the direct use of animal tissues. If someone there, uh, if someone is working with any industries or anywhere in cell culture department, they know the cost of the cells. If we will procure from the NCBI, the 1 microliter costs around 3,500. It is depending upon the type of the cell line and type of the cells. So it is estimated that definitely the cost of production of the cells that is more about 10 times higher than the direct. And the control of environmental factors like pH, temperature, and dissolved gases, and disposable of the biohazards, it is not at all a easy task. So here I wanted to share one my personal experience. When I worked with HCU, by the time I also worked in the laboratory where I have done my experiment, uh, where I have purified a compound called as santalin. So I have checked the capacities, biocapacities of santalin, whether it is a anti-cancer agent or not. During the corona, so we have faced many problems for survival and for the controlling of environmental factors, not only the environmental factors, but also by the time the temperature levels are very high. So because of that reason, I guess the entire unit got blocked, so that our -80 and -20, everything got fired. So we have faced all these conditions. So the native in vivo cells that exist in three-dimensional geometry, while in in vitro tissue culture, the propagation of cells that occur on a two-dimensional substrate. Due to this, the cell-to-cell interactive characters that may get lost. And the cell lines that may represent one or two types of cells from the native tissue, while others may go unrepresented. And the tissue culture techniques that are associated with the differentiation means the loss of characters of the tissues, and from which they were originally isolated. If uh, the point which I have mentioned as a first point, that is expertise and technical skills. So here now I have mentioned the point like differentiation of characters of the tissue cells. So yes, if someone is not expertised in cell culture or the tissue culture, in plant cell we can able to recognize from which plant we have taken the explant. But in case of this culture, we can't able to differentiate if we are not into the subject, because every cell looks like the same if we are not expertise. If it reaches to a stage like a lag phase, lag and log phase, sometimes every cell looks like the same. This is happened to the adaptation and selection process by culturing. So here it is very hard to differentiate and characterize the tissue cells. Here the continuous cell lines that may result in genetic instability. This may ultimately lead to heterogeneity of the cells. The components of the homeostatic in vivo regulations like the nervous system, endocrine system, and metabolic integration that are lacking in vitro cultures, addition of hormones and growth factors that has been started very recently. So when the mammalian cells that divided by the normal and immortal and continuous and transformed. Normal means that divided only for limited of the generations, like 30 generations. And the transformed means that can be propagated. Here we'll see some differences between the normal cell and the transformed cell. Here the normal cell, that is an anchorage dependent cell, except the blood cell. Here the transformed cells, that is non-anchorage dependent, like a suspension culture is possible. Here the normal cell, which is immortal, there will be like a finite number of divisions. Here the transformed cells are the more immortal or continuous cell lines. And normal cell lines that contact inhibition. Here you can see the presence of monolayer culture. In a transformed cells, no contact inhibition. There you can see the multi-layer culture. Yesterday we have covered this monolayer, multi-layer words. So dependent on external growth factors, signals for proliferation. Here the transformed cell that may not need any external source of growth factors. Here the normal cells that greater retention of differentiated cellular function. Whereas transformed cells that typically loss of differentiated cellular function, and normal cells that display the typical cell surface receptors, whereas transformed cells, the cell surface receptor displayed may be altered. So we already discussed about the major developments in the cell culture technology, that with the first development that is using of the antibiotics that which inhibits the growth contaminants, and the second was after the using of trypsin to remove the adherent cells to subculture further from the culture vessel. So third was the use of culture vessel. Sorry. Third was the use of chemically defined culture medium. So here a growth medium or the culture medium is a liquid or gel that is designed to support the growth of the cells. Here we have two types of culture media, cell culture media, that is a natural media and artificial media. Here natural media, it is a type of media like biological fluids. Here the biological fluids like plasma, serum, lymph, human placental cord serum, amniotic fluid. And the tissue extracts, here the extracts of liver, spleen, tumors, leukocytes, and bone marrow, and the extracts of bovine embryo and chicken. And the clots like coagulants or plasma clots. When coming to the artificial media, yesterday we already discussed about MEM and DEM, and what is MEM and what is DEM, and RPMI also we discussed, right? So here the artificial media means the balanced salt solutions. Here the balanced salt solutions are PBS, DPBS, HBBS, HBSS, and EBSS. Along with that, basal media like a DMEM media, MEM media, and complex media like RPMI 1640, IMDM. All these are the media where we use all these material like artificial media in our regular laboratories. So natural media, yes, it is very useful, and lack of knowledge of the exact composition of these natural media, it is failed to use sometimes. So in this type of media, we use tissue extract, liver, and spleen, bone marrow, chicken embryo extract, and other body fluids like amniotic fluid and lymph fluid. And when coming to the artificial media, serum containing media, serum free media. Even in the last slides, I also mentioned that storage of cells. Here, serum free media is used for storage of cells. So here it is a defined culture media and chemically defined media and protein free media. So artificial media, it is a defined media, which is basic benefit of this media is that all defined nutrients, but pH of this should be always 7. So the purpose of type of media that is using which provide all known nutrients to the cell. We also can include some other nutrients which cause cell proliferation and differentiation. So serum free media, in this media, using the serum as a nutrient base for cells, and benefit of using serum free media is we control to cell growth and also include cell proliferation factor and differentiation factor in it. But This has also having the disadvantage like uh, slow growth rate, and that requires the reagents totally pure condition. So serum media, it is one of the example of natural media. Like role of this media in the cell culture is very complex. That also has some growth inhibitors which affects to the cell culture, and serum that helps for cell attachment and work as a spreading factor in the culture. This work add in the cell culture like culture albumin carrier factor that help in reduce to mechanical damage, and also work as a natural buffering agent that also help to maintain the pH. Here pH of the medium always should be like a 7. The basic components of the culture media is the culture media as a powder, as a liquid. Nowadays, the artificial media is not at all in powder. Previously, we used to uh, use the powder media. So, for uh, problems with the contamination, we are using the artificial media, the one which contains amino acids, glucose, salts, vitamins, and other nutrients. The requirements for these components that vary among the cell lines, and these differences are partly responsible for the extensive number of medium for formulation. Like uh, yes, if someone wanted to grow HeLa cell lines and HEPA cell lines and normal cell lines, we use DMEM media. For someone wanted to grow breast cancer cell lines, uh, they have to use RPM. Right? But anyways, DMEM gives the proper growth. If we wanted to check their molecular levels, so definitely we will go with the RPM media. So definitely the requirements of the culture media depending upon the cell. So here this media is also containing natural buffering system, HCPS, and phenol red. Here the phenol red is the pH indicator. Means the culture media is when we are passaging the cells, the culture media is in pink color only. If we will start monitoring the cells, soon after we will inoculate it. With every 12 hours, yes, definitely we can see the changes in the media, because uh, here the phenol red acts as a pH indicator. If it is turned into yellow or the purple, yes, definitely the being pH you can identify. Once the cells reach to the stage like stationary phase and near to decline phase, it is totally becomes like yellow color. So inorganic salts and amino acids and carbohydrates, proteins and peptides. Here the proteins and peptides, it is important in serum-free media. Serum is a rich source of protein, and it includes that like albumin, transferrin, eprotidine, putin, and like fibronectin, and the fatty acids and the lipids, the vitamins, and some small traces elements. So the common cell culture media, like already I told you about EM medium means Eagle's minimum essential medium. EM media means Dalbacco's modified Eagle's medium. It is a modified Eagle's medium. How it is modified means it is like low glucose and high glucose. So called as EM medium, and RPMI medium, Ham's nutrient mixtures, and IMDM means ISCO's modified Dalbacco's medium. And the criteria for selecting the media is the immediate survival and the prolonged survival and indefinite growth and specialized functions. It's always good to start with the MEM medium, means minimal essential medium, for if someone wanted to expertise after preparing the medium. So go with the MEM, minimal essential medium. So for adherent cells and RPMI 1640 for suspension cell. Here uh, hope now you can see my. Yes, because here I am getting that my screen is sharing. So here, just now I used the name like a HeLa cell lines. Oh, thank you. So here the HeLa cell lines, the epithelial cell lines, which is originated from the human. For that, we need MEM media and glutamine and FBS and 1% of non-essential amino acids. So what is the applications of after using this cell line means we can able to check the tumorigenicity and virus studies. As like that, here we have different types of cell lines. HEK 293. We also have HEK 273 for transformation studies. I used this HEK. It is very easy to grow HEK cell lines after using the DMEM medium and EMEM medium. So yeah, here are the mediums which we can use for the growing of these cell lines. For expression and virus replication studies, we use COS7. I worked on HeLa cell lines COS7 and HEK 293 and HEK 273 also I worked on. So here most of the most of the cell lines which is derived from the human only. Only COS7 is from monkey, and 323 and A31 also from mouse, and CHO from hamster. So here the common media and their applications, just now we discussed. IMDM, here the tissue are the cell lines, which you can so here the DM media we use. So this media which is used like bone marrow studies, hematopoietic progenitor cells, and human blastoid leukemia cell lines. So here these are all the media types and their applications. So here media supplements, we already discussed about the media supplements, the serum in the media, like basic nutrients, growth factors, and binding properties, and promote the attachment of the cells to the substrate, and protease inhibitors, and several minerals like we require the sodium ions and potassium, zinc, and iron. So this protects from the mechanical damages during the continuous agitation during the suspension cultures. So serum media, which acts as a buffer for giving the favorable environment for the cells. So when coming to the organ cultures, before going to this cell culture equipments also we need to study. Like here we need the cell culture equipments. As everyone knowing, we require the incubators. The specific requirement of the cell culture laboratory, that is mainly depending on the type of the research that is conducted. For example, like the needs of mammalian cell culture laboratory that specializing in cancer research is quite different from that of an insect cell culture laboratory that focuses on protein expression. However, all cell culture laboratories have common requirements of being free from pathogenic microorganisms like aseptic conditions. Uh, Here the basic requirements like incubators, that everyone knowing that here incubators should have to provide, should have to have 37 degrees, and it is also have to provide the CO2. So it is also have to circulate the air, that is forced air circulation, and should be temperature controlled to within plus or minus 0.2 degrees, and stainless steel incubators that allow easy cleaning and provide a corrosion protection, specifically in human air, humid air is required for the incubation process. Although the requirements for the aseptic conditions in the cell culture incubator, it is not as stringent as that in cell culture hood. Frequent cleaning of the incubator is essential to avoid the contamination of the cell culture. So here we have the different types of the incubators. Yes, now I told you, no, CO2 incubators and dry incubators. So, and humid CO2 incubators. Here the dry incubators is more economical and but require the cell cultures to be incubated in a sealed flask to prevent the evaporation. So that placing a water dish in a dry incubator that can provide some humidity, and but they do not allow the precise control of atmospheric conditions in the incubator. So the humid CO2 incubators are more expensive when comparing with the dry incubators. So, but allow the superior control of culture conditions. They can be used to incubate uh, the cells cultured in the petri dishes or multi-well plates, which require a controlled atmosphere of high humidity and increased CO2 tension. For storage capacities, here some media and reagents, those were light sensitive, while their normal laboratory used under light condition is tolerated. They should be stored in the dark or wrapped in aluminum foil when not in use. So for storing the uh, liquids such as media and reagents, for chemicals such as drugs and antibiotics, and for some consumables and disposable pipettes, culture vessels, gloves, glassware, media bottles, and media pipettes. All these equipments, all these uh, things we can keep, and glassware, plastic. Uh, yes, definitely we need ambient temperature on shelves in dryers, so that it is important to store all these media, reagents, and chemicals. That is according to the instructions on the label, which is given by the instructions which is given by the companies. So the refrigerator, as just I told you, 4 degrees we require, 2 to 8 degrees we require, minus 20 refrigerator we require, and minus 80. All these we consider like freezers. And cryogenic storage, we have some cryogenic vials, so that we'll keep narrow necked containers and wide necked containers. So we also require the cell counter. A cell counter is essential for quantitative growth kinetics, and a great advantage when more than 2 or 3 cell lines are cultured in the laboratory. So with this, before going to the organ culture, okay, we also discussed about some organ culture. Here the organ culture or the representative fragments with reference to structural integrity and nutrient and gas exchange, both and differentiation. So here the structural integrity means the cell to cell association and interactions found in native tissues or organs are retained to a large extent as the structural integrity of the original tissue is preserved. The associated cells that can exchange signals through cell adhesion or communication. And here the nutrient and gas exchange. There is no vascular system in the organ culture. This limits the nutrient supply and gases exchange of the cells. This happens despite the adequate care taken in the laboratory for the rapid diffusion of nutrients and gases by placing the organ cultures at the interface between the liquid and gaseous phases. As a consequence, some degree of necrosis at the central part of the organ that may occur. Uh, some researchers that feel like high oxygen concentration, sometimes even pure oxygen in the organ culture, which is exposed of cells to high O2 content is associated with the risk of oxygen-induced toxicity. Nutrient metabolite exchange is severely affected, and the growth and differentiation. In general, the organ cultures that do not grow except some amount of the proliferation that may occur on the outer cell layers. Here, the advantage of organ culture is provide a direct means of studying the behavior of an integrated tissue in the laboratory, understanding of biochemical and molecular functions of an organ tissue that becomes. Yes, there are some limitations of organ culture, like organ culture that cannot be propagated Hence, for each experiment, there is a need for a fresh organ from the donor. So it is very difficult to propagate the organ. So variations are high and first reproducibility is difficult to prepare besides being expensive. So the basic technique of organ culture that consisting of the like a dissection and collection of organ tissue. Reduce the size of the tissue as designed and preferably to less than 1 millimeter in thickness that allows to grow in the media. Place the tissue on supported the gas medium interface. Incubate in humid CO2 incubator. Just now I told you, no, humid CO2 incubator. And change the medium. Here we use the medium like M199 or CMRL 1066 as frequently as desired. The organ culture that can be analyzed by the histology, autoradiography, and immunochemistry. Here you can see the dish, the organ culture in the stainless steel supported grid, the small fragment. Here you can see the small fragments of the tissue that can be cultured on the filter laid on the top of the stainless steel grid. Here it is the top position of the grid. So this contains like a dish and a medium. Here you are going to place in the dish. So the organ culture on filter well insert. The filter well which containing it is a popular uh, vessel for organ culture. This is mainly because the cellular interaction, stratification, and polarization are better in this culture systems. And the recombination of cells to form tissue like densities and access to medium and gas exchange that are looks very better in after using this filter well insert. Here you can see the presence of media where you can see the cell layer. So it is the initial growth of the cell. So here we are adding continuously a fresh medium. So this is the monolayer on top of the filter. So monolayer, this is the one which avoids the contamination. That is what we will prefer. That is what this mechanism is become like very popular for organ culture. So here we have the 4 types of different wells for growing the tissues is in the form of cell layers. Yes, that is already depicted in this picture. Like this is monolayer and the monolayer grow on the matrix on the top of the filter. Interactive cell layer on the underside of the filter. The interactive cell layer on the underside of the filter with matrix coating. So here this picture that explains you that the growth of the cell layer on the top of the filter and the growth of the cell layer on the matrix like which is made up of by the collagen or matrigel on the top of the filter and the cell layers that are grown on the interactive cell layers that is placed on the underside of the filter. This filter well inserts with different materials like ceramic, collagen, and nitrocellulose membrane are now commercially available for use in the cultural laboratories. And the filter well inserts that have been successfully used to develop the functionally integrated thyroid epithelium, stratified epidermis, and intestinal epithelium or renal epithelium. There are many success stories for growing organ culture. So, with this, our next topic is stem cell. Yes. Everyone knowing what is stem cell. Here, the stem cells are the cells which is undifferentiated cells that are presented in the embryonic, fetal, and adult stages of the life of the living organism and gives rise to the differentiated cells that are building blocks of tissue and organs. Here, the stem cells that are differentiated from the biological cells that can differentiate into specialized cells or can divide to produce more stem cells. They found in multicellular organisms, as just we discussed, living organisms. In the postnatal and adult stages of the life, the tissue-specific stem cells that are found in differentiated organs and are instrumental in repair or following the injury to the organ. The main characteristics of stem cells are, yes, it is self-renewable, renewal, means the ability to extensive. Okay. The ability, where here? Yes. The ability to extensively proliferate. Yes, the cells can proliferate easily and you say like usually arise from a single cell, that is like clonality, and potency means the ability to differentiate into the different cell types. Here I wanted to introduce one term like totipotency that we will discuss in the later slides also. Means the ability of the cell that divide and redivide is called as totipotency. So these properties may differ between various stem cells. For example, like embryonic cell, stem cell that derived from the blastocyst in mammalian cell. There are two broader type of stem cells like embryonic stem cells which are isolated from the inner matrix of the blastocyte and adult stem cells which are found in various tissues. So the embryonic stem cells that is derived from the blastocyst have a greater ability for self-renewal and totipotency, while stem cells found in adult tissue have limited self-renewal since they would not proliferate extensively and can only differentiate into the tissue-specific cells. Okay, so yes, definitely, which are found in various tissues. In a developing embryo, stem cells that can differentiate into the all specialized cells like ectoderm, endoderm, and mesoderm. Here, the human body that develops from the zygote and the blastocyst from the embryonic stem cell that are derived into the germ cells or germ layers like ectoderm, mesoderm, endoderm, mesoderm, and ectoderm, right? So specific organs that arrive or arised from the germ layers, some of the progenitor cells that have contributed uh, to organ formation, that do not terminally differentiated but are retained as tissue stem cells, and that can be found in bone marrow, bone, blood, liver, muscle, brain, adipose tissue, skin, and the gastrointestinal tract. The tissue stem cells that may be called as progenitor cells, since they have rise to terminally differentiated and specialized cell of tissue or organ. These cells may be dormant, which tissue, but that would proliferate under circumstances of injury and repair. So the dynamics of tissue stem cell or progenitor cells varies from tissue to tissue. So, for example, in bone marrow or liver, lung, and gut, the stem cells regularly proliferate to supplement cells during the normal turnover or the injury, while the pancreas and the heart or nervous system, they proliferate to replace damaged cells which following the injury. So yes, there are some features about stem cells. Here the stem cells that are unique, very unique cells, stem cells that are having amazing ability to develop into the several distinct cell types that we already just discussed. And the stem cells can be defined or used as a repair system for the body. The stem cell that can divide without a limit in the living organism in order to replenishing various types of cells. When a stem cell that divides, each new cell has the potential to either remain a stem cell or become another type of the cell with a more specialized function like a muscle cell, a red blood cell, a brain cell. Just now we discussed. Before going to the classification, yes, we go with classification depending upon the origin. Here we have the stem cells from embryos, stem cells from the fetus, and stem cells from the umbilical cord, and stem cells from the adult. So yes, just now we discussed about self-renewal capacities of stem cell, the ability to go through numerous cycles of the cell division while maintaining the undifferentiated state, a potency, the capacity to differentiate into the specialized cell types. Yes, self-renewable means the renewal is the process by which stem cell divide to make more stem cell. The self-renewal is division with maintenance of the undifferentiated state. Each stem cell has the potentiality to remain as a stem cell or differentiate to another type of cell through differentiation. Here the potency, before going to the potency, here I wanted to give a small description about the stem cell. The idea of wound repair and organ regeneration, it is as old as humanity and is reflected in the ancient Greek myth of Prometheus, the Greek titan punished by the Zeus. I'm sorry if the pronunciation is wrong, Zeus, Zeus, for his disobedience in introducing the fire and knowledge to the human beings. So yes, it is definitely it is a myth actually. It is a myth, it is a story. So the Prometheus is tied to a rock, and every day as he eats the part of his liver, which then regenerates, In the modern day medicine, the work involving stem cell and organ regeneration that commenced with the first attempts at bone marrow transplantation in animal models in 1950s. These pioneer studies paved the way of human bone marrow transplantation, a therapy now widely used in various blood disorders. This new strategy that revealed the existence of stem cells that regenerated adult tissue. Presently, the regenerative medicine is a major focus of research, not only to find out the therapy, but also to understand the basic biology and pathogenesis of the diseases. So, although there are a number of ethical issues that have arisen in the stem cell research, recent advances in the stem cell isolation and development have helped the scientists, many scientists, to identify and culture the specific cell types for regeneration of tissue in various disorders as Parkinson and Alzheimer's, or diseases of heart, muscle, lung, liver, and other organs. So here the stem cell classification, the potency specifies the differentiation. Oh, now you can see clearly. So one of the main characteristics of the stem cell that varies between the stem cell, that depending upon their origin and their derivation, all stem cells that can be characterized according to their differentiation potential, with about 5 groups like totipotent or omnipotent, pluripotent, multipotent, oligopotent, and unipotent. So here the totipotent, here you can the fertilized egg that divides and redivides. The unorganized cells of cell that is derived from the cell is called as totipotent or totipotent. It is also called as omnipotent. And pluripotent, here the pluripotent cells from the totipotent cells, blastocysts that containing the pluripotent stem cell, here you can see, and isolated pluripotent from the inner cell mass. These pluripotent cells, here the origin for the pluripotent cells are embryo stem cells, and multipotent cells are from fetal stem cells, and oligopotent cells that are from adult or somatic stem. So here the totipotent cells from embryonic extra embryonic tissue, pluripotent cells from 3 germ cells, while multipotent cells that generate cells limited to the 1 germ layer, and bronchoalveolar duct junction cells in the lung may be multipotent, while type 2 pneumocytes are oligopotent and differentiate into the type 1 pneumocytes of the alveoli. Here the totipotent cell or omnipotent cell that are most undifferentiated cells are found in early development of fertilized egg and fertilized oocytes. And the cells of two divisions are totipotent cells. They differentiated in both embryonic and extra embryonic tissue. There may be formation and embryo and the placenta. When coming to the pluripotent stem cells are able to differentiate into the cells that are arise from the three germ cell layers that we already discussed: ectoderm, endoderm, and mesoderm. Here, the pluripotent stem cells that called embryonic stem cells were first derived from the inner cell mass of the blastocyst. Recently, Takahashi and Yamanaka generated pluripotent cells by reprogramming somatic cells. These cells are called induced pluripotent cells and share similar characteristics with embryonic stem cells. Notably, there has been no pluripotent cell population isolated to the lung. Multipotent cells. It is like multipotent stem cells are found in most tissues and differentiated into cells from a single germ layer, mesenchymal stem cells, and the most recognized multipotent cell. They can be derived from okay, ma. Okay, so they can be derived from a variety of tissues that include bone marrow, adipose tissue, bone, umbilical cord blood, and peripheral blood. So here, mesenchymal stem cells that are adherent to the cell culture tissues and characterized by the specific surface cell markers. Very recently, mesenchymal stem cells that were differentiated into a neuronal tissue, which is derived from the ectoderm. This is the example for trans differentiation. We'll discuss all these in the latter classes. And the other cells are oligopotent stem cells. Oligopotent stem cells are able to self-renewal and form two or more lineages within a specific tissue. For example, like ocular surface of the pig, including the cornea, that have been already reported to containing the oligopotent stem cells that generate individual colonies of the corneal and conjunctival cells. Hemopoietic stem cells are a typical example of oligopotent stem cells. And they can be differentiated into both myeloid and lymphoid lineages. In the lung studies suggested that branchial alveolar duct junction, that cells that may give rise to branchiolar epithelium and alveolar epithelium, and unipotent cells. Finally, unipotent stem cell that can have the self-renewable capacity and differentiated into only one specific cell type and form a single lineage, such as muscle stem cells, that gives rise to matured muscle cells and not any other cell. In the lung, type 2 site of the alveoli that gives rise to type 1 pneumocyte. These are the examples for the unipotent cells. So, here you can see the embryonic stem cell structure. We'll discuss all these in the next class. So, with this content, here I am signing off. Thanking you all for your attention. So, tomorrow we will discuss the same topic, the stem cell, followed by cellular aspects of tumor cells, and in vitro fertilization also will try to complete the block 1 tomorrow. Okay, ma'am. Thank you.
Thank you, ma'am.
ఏం కాదు మేడం అన్ని గీసి పెట్టుకుంటాం ఏం కాదు క్యారెక్టర్ గీసినాను
టూ సైడ్స్ రావాలి కదా
ఓకేనా
బ్యాక్ సైడ్ కూడా సేమ్ అలాగే అది దాని మీద తప్పిస్తాం కదా షీట్ మీద
మళ్ళీ ఆ షీట్ మీద చేయాలి ఓకేనా
షీట్ మీద ఇంకో పేపర్ తప్పిస్తాం కదా ఇవి ఇంకొక లెక్క టూ టూ చేయాలి
నేననే చెప్తున్నా
పిలిచిన ఇప్పుడు పచ్చేసి దాని మీద పెడతాం కదా అవి రిపోర్ట్ కలర్ దాంట్లో వదిలేసేయండి
మేము చేసేస్తున్నాం అట్లా
పెడతాం కదా కార్డ్బోర్డ్ మీద పెడతాం కదా ఇక పెడితే ఆగట్లేదు కానీ అన్నారు వాళ్ళకి పెడితే బాగాలేదు కార్డ్బోర్డ్ పెడదాము అని ఎవరితో ప్రశ్న చేసుకోవాలి
డబల్ పని అవుతుంది పని కాదు తొందరగా
అలాగే పెట్టేద్దాము పేపర్
లెటర్స్ ఆగవు లెటర్స్ కి పెట్టుకోండి వీటికి ఏమవుతుంది
స్టక్ అవుతుంది చూడు.
ఏదైనా అంతే అవుతుంది
కట్ చేస్తాం కదా ఏముంది ఇప్పుడు ఇదేంటి రన్ అవుతుంది కదా
350 పేపర్స్ పడతాయి 350 పేపర్స్
లైవ్
టర్న్ అయింది కదా.
అయిపోయింది కదా
హెచ్పి
పెట్టి 4 నెలలు అవుతుంది ఎప్పుడైపోతుందో తెలియదు.